So, hello everybody. Today is my pleasure to introduce Dr. Sergio Gradilone, who is Associate Professor and Section Leader of the Unit of Cancer Cell Biology and Translational Research of the Hormel Institute at the University of Minnesota in Austin, USA. I knew Sergio long time ago when we were at the University of Rosario in Argentina. He was doing his PhD in biological science, and I remember that he was studying the aquaporins in the liver. Then I was doing there my thesis for the master degree. And he was studying this aquaporin that was very interesting for me at that time. Then Sergio moved to the Mayo Clinic at Rochester to continue his and develop his career in the United States. So today Sergio will present to us some of the novelties regarding his studies on the role of the primary cilium in health and disease. So Sergio, when you want, you can start.
Fantastic. Thank you so much, Pablo, for the introduction, very kind introduction, and thank you everybody for the invitation and the opportunity to share with you our work. I see if I can share the screen now. Can you see that?
Yes, we can. I will close my microphone now.
Perfect. So as Pablo just mentioned in the introduction, the focus of our lab is we study the role of primary cilia in tumor biology, in disease in general, and inside the liver, of course, we study the biliary cells, the cholangiocytes. For this audience, I don't need to spend time here. This is just a 3D reconstruction of a rat liver lobule, where you, of course, see the hepatocytes in brown. And inside the liver, we found the biliary tree. These are very important cells, even though they are a minority in the liver. They have a very important physiological role in the generation of bile. So we study these epithelial cells known as cholangiocytes. And these cholangiocytes that are involved in the formation of biles are the ones that express this primary cilia that is like a cellular antenna that is sticking out of the plasma membrane into the environment, sensing what's going on in the environment. We can isolate cholangiocytes and grow them in vitro, and we have antibodies to stain ciliary markers, and you can see in red there how the primary cilia is pointing up to the media, sensing what's going on in the environment. Cilia has been known forever, and the most common probably that you are familiar with are the motile cilia, that you will find them in the respiratory tract, in the reproductive tract, and they are usually hundreds per cell, and the function is to move the liquid that is over the cells. You also find motile cilia in the sperm cells and in some unicellular organisms like Chlamydomonas. And then we have the primary cilia or non-motile cilia. This is normally one per cell, and they are found almost in every cells of our bodies. The main difference between these two is the ultrastructure. So motile cilia has two microtubules in the center of the axoneme that give the mechanical force with the interaction with the external microtubules to move the structure. On the other hand, the primary cilia is lacking the central pair. So the ultrastructure is now 9+2 because there are 9 pairs of microtubules plus 2 pairs in the motile cilia and 9+0 in the non-motile or primary cilia. This is an immunofluorescence of transversal cut of a bile duct. We were very lucky here to find a huge cilia in the lumen. So these are the nucleus of the cells forming the bile duct. And this is a very nice primary cilia in cholangiocyte primary cilia. As I mentioned, if we take a look inside, is the ciliary axoneme is conformed by 9 pairs of microtubules. And remember this, in mammalians, the microtubules are acetylated, which confers them the stability to keep the structure up and working. Primary cilia in the cholangiocytes were described for the first time in 1963. And I always like to show this, the abstract of this original paper, where the conclusion is that they may represent embryological remnants without the function. And that was the view of primary cilia at the time, just evolutionary junk without the function. Then in 1989, Nick Larusso's lab at Mayo Clinic published this image of primary cilia in the cholangiocyte, but just as an anecdotic point in the paper, no more description of that. And it wasn't until 2006 that we started to understand the function of these organelles. And I was super lucky because I was just arriving to Larusso's lab for my postdoctoral training at Mayo Clinic. This is a scanning electron micrograph of a rat liver, where you can see the portal vein, the hepatic artery, and a bile duct. So if we zoom in there, you can appreciate the primary cilia, these hair-like structures inside the lumen. So this is, I mean, you can imagine bile is flowing there. So primary cilia are ideally positioned to interact with luminal bile. And over the years, we demonstrate that they can work as sensing the flow, the composition, the tonicity of bile, and sending signals inside the cell. For the mechanosensation, of course, this function depends on the specific expression of proteins that are enriched or specific to primary cilia. For mechanosensation, the polycystins are important, polycystin 1 and 2. For chemosensation, primary cilia can detect nucleotides expressing purinergic receptors, can detect bile acid by expressing TGR5 there. They also can bind exosomes. And they can also sense osmolarity, changes in osmolarity in the liquid by the expression of a calcium channel known as TRPB4. And of course, primary cilia are not only important for normal physiology, but the boom in the study of primary cilia started when it was discovered that genes that were causing polycystic kidney disease were related to the expression of primary cilia. And from there, a ton of different diseases now known as Ciliotherapies were discovered, including diseases in the liver, like polycystic liver disease, for example. But of course, when I was finishing my postdoc and looking for my niche in research for my independent career, we decided to study what was the fate of primary cilia in tumor biology. And obviously, we studied cholangiocarcinoma, the tumors that derive from the cholangiocyte. So this is a very important malignancy, even though it's not that common, it's very aggressive. So we have three variants of these: intrahepatic, peridistal, perihilar, and distal cholangiocarcinoma. Depends on the anatomy of the location of the tumors. The incidence of this tumor has been increasing worldwide over the decades. And as I mentioned, it's a very aggressive tumor with a median survival of only 24 months. And there are not good treatment for this beyond the palliative chemotherapy treatment. So what we found is that primary cilia are usually lost in tumors. this is an immunofluorescence in a normal liver showing a bile duct staining in green for CK19 and in red for a ciliary marker. In this case is acetylated alpha-tubulin, as I mentioned before. The microtubules of the primary cilia are acetylated in mammals. And in the tumor samples, primary cilia are usually gone. And we were also able to reproduce these in vitro. So when we compare normal cell lines like the 869 or the NAC, normal cholangiocyte cell lines, we can stain very nice cilia. In red here, in green is the basal body of the cilia, staining with an antibody against gamma tubulin. And in the tumor cells, primary cilia are very hard to find, even though we grow the cells in the conditions that maximize the ability of the normal cells to produce cilia. So we were, of course, the question was, is this important? It's just a characteristic of tumor cells. Is this important or not? So the first approach was very naive. We just took normal cell lines and removed the cilia by different approaches. So we can use a chemical deciliation using the drug chlorhydrate, even though the mechanisms are not well described. There is evidence that removes cilia. And of course, using some molecular approach, using knockdowns or knockout of proteins that are important for the process of ciliogenesis, like IFT3A or KIF3A, for example. And then we assess the phenotype of the cells. So what we found is that when we remove cilia from normal cells, we see hyperproliferation of the cells. So the cells start to grow more. Not only that, they have more migration. This is the same cell line with and without cilia, and we see increased migration. There is increasing anchorage-independent growth, increasing invasion, and increase in signaling pathways that are usually related to cholangiocarcinoma, like MAP kinases, in this case by staining phospho-ERK, Hedgehog signaling pathway, interleukin-6, decrease in p53. So very interesting phenotype when we remove primary cilia, suggesting that the primary cilia may be working as a tumor suppressor organelle. Now we have done a genetic approach in mice to try to support this hypothesis. So what we did is to generate a mice that has a liver activation of KRAS and a knockout for IFT88, so we don't have cilia in the cholangiocytes, as shown in this immunofluorescence, wild-type mice with the primary cilia in red, no primary cilia in the double knockouts. And what was very interesting is that KRAS activation alone has very low penetrance to form tumors. But when we combine that with the loss of primary cilia, we have an increase in the generation of tumors in these mice, supporting the idea that primary cilia may be working as a tumor suppressor organelle. So we came out with this idea of Ciliotherapies and the general idea that normally cholangiocyte primary cilia, the multisensory functions of these organelles, are inhibiting tumor promoter signaling pathway or maintaining the expression of tumor suppressor signaling pathway, acting as a tumor suppressor organelle as a whole. And we propose a Ciliotherapies approach to cholangiocarcinoma, and we have two ways to go with this. One is to rescue the expression of primary cilia in the tumor cells, and another way would be to mimic the functions of primary cilia as a therapeutic approach. So to rescue the expression, we need to understand the mechanisms of ciliary loss that are used in the tumor cells. And to mimic the functions, we need to understand what is the signaling pathway or what are the signaling pathways that are activated upon ciliary stimulation that may regulate growth and migration and invasion. And then, of course, when we have something interesting, we test this in animal models and moved to clinical trials one day. Regarding the mechanisms of ciliary loss, what we found is that the deacetylation of the ciliary axoneme is very important and critical in the tumor cells to remove the cilia. As I mentioned before, the axoneme, the tubulin in the axoneme is normally acetylated. So tumor cells are overexpressing deacetylases to make this unstable and remove the cilia. We found by now, for now, we know at least two of them, HDAC6 and SIRT1, are involved in this mechanism. So basically what's going on is that these deacetylases are targeting the microtubules and inducing the resorption of primary cilia and helping the malignant phenotype. So we have done a lot of work on HDAC6 overexpression in cholangiocarcinoma, showing the role in ciliary loss. This immunofluorescence showed the overexpression of HDAC6 in cholangiocarcinoma samples, and we play a lot with this. We overexpress HDAC6 in normal cells that induce decreased acetylated alpha tubulin levels and decreased levels of primary cilia in the normal cells. And that correlates with an increase in proliferation of the normal cells, as I showed you before, and an increase in anchorage-independent growth. On the other hand, if we take tumor cells and knock down the expression of HDAC6, in this case using shRNAs, we have increased acetylation of alpha tubulin. We now start to see primary cilia in the tumor cells, and this correlates with the decrease in proliferation of the tumor cells and decreased activation of the signaling pathways that I showed you before related to CCA. We can do a pharmacological approach to this, so there are specific inhibitors for HDAC6 now available. This is an experimental one, tubastatin-A. When we treat tumor cells with tubastatin-A, in this immunofluorescence, confocal immunofluorescence, we can see primary cilia starting to express in the cell surface, compared with no cilia in the tumor cells per se. And again, this correlates the treatment with tubastatin-A decrease the proliferation of the cells, and does that in a ciliary-dependent manner. This is very important to know. So what we did is we stably transfect these cells with an IFT88 shRNA. So these cells are not able to grow back cilia after the treatment with tubastatin-A. And what we see is that the effect in proliferation is almost abolished. So the regrowth of primary cilia is a condition for tubastatin-A to inhibit the proliferation of these tumor cells. And the same for colony numbers when we did the soft agar assays. So the idea is that these deacetylases are like big wrecking balls that are destroying the primary cilia and favoring the malignant phenotype of the cells. We test this tubastatin-A in cholangiocarcinoma, a rat model. We use an orthotopic model developed by Dr. Sirica lab. So basically we are injecting tumor cells in the liver and assessing the growth of the tumors, and tubastatin-A reduce the growth, and that correlates with the appearance or with the expression of primary cilia in those small tumors that we are finding. This approach of HDAC6 moved to clinical trials. I mean, we put everything together, it's almost there, but of course, always a funding problem. So they didn't start yet the trials, but it's a work in progress. We moved forward studying this, then we discovered that autophagy is also involved in this resorption of primary cilia in a process that we can call ciliophagy. So HDAC6 and autophagy work together to bring all the ciliary components to the autophagy machinery. And as I mentioned before, now we know also that SIRT1 is also involved in the resorption, work done by these postdocs in the lab, Adrian, Stanislao, and Kishore. This is just simple showing you a recent paper showing the overexpression of SIRT1 in tumors. This is a tumor tissue microarray of cholangiocarcinoma from Mayo Clinic, showing the upregulation of SIRT1 in the cholangiocarcinoma. This is a different cohort from Italy, collaborators in Italy, showing that SIRT1 is expressed in the tumors compared to normal areas or even surrounding tissue. And then when you go to the TCGA public database, also overexpression of SIRT1 in cholangiocarcinoma. So again, we play here overexpressing, well, this is just cell lines models, sorry, SIRT1 overexpression in tumor cells, 4 different tumor cells compared to the 2 normal cell lines that we have overexpression of SIRT1, supporting the idea. So again, we play with the overexpression of SIRT1 in normal cells that correlate with decreased levels of acetylated alpha-tubulin and decreased expression of primary cilia in both normal cell lines. The opposite side of the experiments, we knock down SIRT1 using shRNAs in tumor cells or using a specific inhibitor, sirtinol, to inhibit the activity of SIRT1. And again, we start to see increased expression of primary cilia in these tumor cells. And this again correlates with changes in proliferation. So when we overexpress SIRT1 in normal cells, we see hyperproliferation. When we knock down the activity by shRNA or sirtinol, we have decreased proliferation in the tumor cells. And as I show you again for HDAC6, in this case, SIRT1 is effect on proliferation depends on the ability of the cells to regrowth primary cilia. So this is a cholangiocarcinoma cell line showing decreased proliferation with an siRNA for SIRT1 or using sirtinol. And when the same cell is transfected with an shRNA for IFT88, so these cells are not able to form primary cilia, there is no much effect of these inhibitors on proliferation, suggesting that both the inhibition of HDAC6 and now the inhibition of SIRT1 effects on proliferation in these tumor cells depends on the ability of the cells to re-express primary cilia. We also did animal models showing the effect of sirtinol here, reducing the growth of the tumors and affecting some of the signaling pathway that we are studying. Very interesting, butyrate, this short chain fatty acid that is the production in the gut can be regulated by diet and by the microbiota. Was described to inhibit SIRT1. So we are trying this as a potential intervention in the future, a dietary intervention in the patients, showing that butyrate has mostly no much effect in the normal cells, but is able, of course, to induce the expression of primary cilia in the tumor cells and decrease the activity of SIRT1 in both tumor cells. And when we measure proliferation, that was very interesting. There is a dose response in both tumor cells, decreasing proliferation of the cells, but there is almost no effect. We start seeing the effects at higher dose in the normal cells. So that's always encouraging when you see effects in the tumor cells and not much effect on the normal cells. So this is a work that hopefully can translate to the clinic using some dietary interventions. So the other side of the approach, consequence of ciliary loss, how we mimic the function. So we are trying to understand all the different signaling pathway regulated by cilia. So Adrian did a nice work showing that chemosensation of primary cilia normally regulates migration and proliferation in the cells. Basically, what we show in that paper is that P2Y11 is normally expressed and localized in the primary cilia in cholangiocytes and can detect nucleotides that transduce the signal using LKB1 as a hub, and that ends up regulating the F-actin network and regulating migration and proliferation. In tumor cells, there is loss of primary cilia, so we are losing this communication between the environment and the cell interior, and we are losing the break here by losing activity in LKB1. So we have increased AKT and increased F-actin network and increased migration and proliferation in the cells. One way to mimic the function is to activate, in this case, LKB1. Adrian did that by using this semi-natural compound, HNC is aspiridine methylchalcone, that can activate LKB1 and reduce migration and proliferation. And we are now heavily studying the regulation of EGFR by cilia and how primary cilia affect the cell metabolism. Those are two very active line of research right now in the lab. Kishore is working on that. So I show you some data on the EGFR story that we are wrapping up. So one approach we did was in vitro using normal cells that we can knock out primary cilia by downregulating different genes. In this case, I show in IFT88, we can do KIF3A, different proteins. And then we did RNA-seq in the search of what are all the signaling pathways that are being affected by the loss of primary cilia. And one of them was EGFR signaling. It came up as a hit. And then when we went to test this at the protein level, we see that EGFR signaling is increased in the normal cells that do not have cilia, here showing IFT88 knockdown, and chloride hydrate is the chemical deciliation. Both cell lines show a similar pattern, increased EGFR. Here in panel B are rat cells. These are normal rat cholangiocytes, and cholangiocytes derived from a ciliopathy model is the PCK rat that actually has a very strong phenotype of polycystic liver disease, and we know that have a very distorted, short and malformed cilia. So it's known as a ciliopathy, and increased EGFR signaling there. We use the mouse model I showed you before, knockout of IFT88 in the liver using an albumin cree. So no primary cilia here, acetylated alpha-tubulin in the bile ducts, and these mice are similar to the PCK rat, so they are cystic livers, and those liver has increased EGFR signaling. The same on the rat, as I mentioned, the PCK rat, the model of polycystic liver disease, increased EGFR signal. So all these data are suggesting that problems with primary cilia are linked to overexpression of the EGFR signaling pathway. And that's actually is not that novel because, of course, EGFR hyperactivation is known in cholangiocarcinoma, has been well described, and there is also evidence that is a problem in polycystic kidney and liver diseases. So what we wanted to understand if there was any relation, any regulation coming from the cilia to the EGFR signaling pathway. So we started to explore this mechanism of EGFR, increased EGFR activity in the ciliary-defective cholangiocytes. So first of all, what we found is that the normal cells that have knockdown of the ciliary genes, so do not express primary cilia, are way more sensitive to the ligand EGF. So as you can see here, the response, I think this is 30 minutes incubations. So huge response compared to the normal cells. And something similar happened in this rat cholangiocytes, normal compared to PCK cholangiocyte. The cells that have ciliary prominence are more sensitive to EGF. We double-checked that using immunofluorescence for phosphoric, again, finding that the non-ciliated cells are more sensitive. They have more response to the EGF treatments. So then we did a time course here on the signaling after EGF treatment. And the observation is that the normal cells that do not have cilia or have ciliary defects have longer time, increase a signal over the time. So in the normal cells, there is a peak at 10 minutes and then the signal is gone. But in the deciliated cells, we have more sustaining signal. So maybe a problem with the clearance of EGFR, activated EGFR in these cells. And what we found super interesting was that normal cells that do have primary cilia, when we treat the cells with EGF, what is happening is that the receptor is translocating to the primary cilia. So we see enrichment of EGF in the primary cilia after treatment with EGF. And that translocation depends on the activation. When we use inhibitors of the EGFR receptor activity, we don't find them in the primary cilia. The same in the rat cells. The EGF, the phospho-EGR is found in the primary cilia after treatment with EGF. So we started to think that upon stimulation, the receptor is translocating to cilia. This can happen through a lateral movement from the apical membrane to the primary cilia membrane, or through an endocytosis process and relocation to the primary cilia. So we tried this endocytosis inhibitor, dinosaur, and what we found is that when we treat the normal cells with dinosaur, the activated EGFR remains active in the membrane. In the deciliated cells, we don't see much difference. It's already super active, so there is no difference with the dinosaur. And basically what we are seeing is that inhibition of endocytosis is precluding the active EGFR to move to the primary cilia. As we can see here, a normal cell, when we treat with EGF, the receptor goes to the primary cilia. When we treat with dinosaur, the receptor remains in the membranes of the cell. So we are starting to think that this translocation is important for the degradation and the termination of the signaling pathway. We did some experiment with proteasome inhibitors like MG132 and PIR41. And again, what we found is that when normal cells are treated with these inhibitors, the activity of the phospho-EGFR remains active for a longer time, similar to the deciliated cells. And when we analyze that by immunofluorescence, when we treat the cells with the proteasome inhibitors, we see that there is more accumulation of the receptor in the primary cilia, suggesting that these active EGFR are being removed from the cilia by a proteasomal degradation process. So this is a little bit the working model we have now in normal cholangiocytes. Upon activation of EGFR by the ligand, the receptor is translocated to the cilia, and from there is targeted for degradation and clearance. So in cells that do not have cilia or have ciliary defects, like in cholangiocarcinoma or polycystic liver disease, we are missing the primary cilia, and we have sustained signal because they cannot be degraded as efficiently because they are not going to the primary cilia where they are targeted to degradation. So as I show you, we have these two strategies here on the mechanism of ciliary loss. We have a very nice story on the HDAC6 inhibitors and SIRT1 inhibitors. So we have identified novel targets that may be used for cholangiocarcinoma therapy, like this specific HDAC6 inhibitor, hydroxychloroquine. We have very nice results with that, and with a synergistic effect with the ACY1215. We are working in the, there is a story that I didn't tell you, but relates to the overexpression of HDAC6 and microRNAs and defects in the expression of these two microRNAs and exportin 5. So we are working in an approach to try to target specifically cholangiocyte with AAVs. We have a colleague here expert in EAVs, so we are trying to develop these AAVs specifically targeting cholangiocyte to express microRNAs or even restore the expression of exportin 5. We have developed a dual inhibitor. We call this Ciliomax. It's a dual inhibitor that targets HDAC6 and the autophagy machinery. So this is work in progress. Again, very nice effects in vitro. We had to test this in vivo now. And as I show you, sirtinol is also promising. On the consequence of ciliary loss and how to mimic the function, we show you LKB1 activation is a possibility. We are now, as I mentioned, working on EGFR regulation and the role of primary cilia in the glucose metabolism, epigenetics, and DNA repair. So trying to give you here a broad view of what we do in the lab, with the hope that some of you may get some interest and we can connect and start slowly with the collaboration. So let me just finish by the usual acknowledgement. People in the lab that has done all the work that I show in today, Seth, Kishore, and Stanislao. Mostly Adrian also did many of the initial work. Collaborators at Mayo, Larusso's lab, and Luis Roberts, Chus Bañales in San Sebastian, Brunella Franco in the TGM there in Naples, and Andrea Balavio. And of course, funding the Hormel Institute that has very generous funding, and NCI with R01 funding, R21 funding. Thank you so much for your attention, and I'm happy to respond and answer any questions.
Okay, thank you, Sergio. It was really interesting, your presentation and all the data that you showed to us to share with us. So we can, if somebody have questions, Sergio is ready to take some of them.
Yes, I have a question. Sergio, first of all, congratulations. It was really, really exciting and well presented, logical, challenging. I have a question. I mean, I think fascinating the idea that celiac may be related to the development of cholangiocarcinoma. This makes a lot of sense. The question I have is a very basic one and very stupid one probably, but I think that clinically speaking may be something important. I understand that the loss of cilia may be related somehow in the development of cholangiocarcinoma. But the question is, let's assume that I can be able, as you mentioned and you show us, to re-express cilia in the cholangiocarcinoma. Is already a developed cholangiocarcinoma. Do you expect the cholangiocarcinoma may be reduced in terms of growth? Or so it's a sort of palliative treatment, in your opinion, or maybe a sort of definite treatment? Because we all know that cholangiocarcinoma is a very aggressive, a tumor with very poor prognosis?
Yeah, that's actually a great question, Claudio. So I would say, based on our data from in vivo models, we started treatments trying to restore the expression of primary cilia when the tumors are already established. And what we see is that there is a huge difference in the growth of the tumors. So we'll stop the growth for sure. If we'll reduce regression, I cannot tell based on the experiments we have, because we haven't done yet the right experiment where we have a measurable tumor and we can see if it regress. That's on the plans, but we didn't do that yet. My speculation, my hypothesis is that that will be the case, because these cells, when are able to restore primary cilia, will find an environment that the cilia will signal inside the cell that will likely induce cell death. So my hypothesis is that this will also help regressing the tumors.
Well, this is going to be fantastic, because this is going to be the real, resolution of something. But also, if you freeze the growth, that could be helpful in order to give more time to prepare a liver surgery or a transplantation.
Yeah.
That's good. Very, very nice, Joe. Thank you.
Thank you.
Mil gracias.
De nada, un placer.
Other questions? If not, I have one. Maybe I lost some information, but regarding the stimulus that cause this loss of the cilia, there are information about this. What the stimulus can do that, or if there is some, you think that the pathocyte is sending some signals to the cholangiocyte, or what information is?
So
exists?
Let's see if I can follow your question. If you are talking in physiological conditions, yes, definitely signals. Normally, what primary cilia in a normal bile duct is doing is sensing what's going on in bile. So in the bile. So that's can be nucleotides or exosomes or bile acids secreted by hepatocytes or by upstream cholangiocytes that are signaling downstream cholangiocyte, and that will fine-tune the ductal bile secretion. For example, if the mechanosensory function, when you have increased flow, so there is increased production of bile, the primary cilia sense that and end up sending a signal to inhibit the secretion of bicarbonate and water by the cholangiocyte. So trying to stop the flow. Something similar happened with osmosensation. Yeah, it's a crosstalk between hepatocytes and upstream cholangiocyte with what's going down.
And so something is activating this deacetylases that you are studying practically.
Yeah, that's
one of the.
Yeah, in the tumors, we know that it's the overexpression of the deacetylase that are inducing the loss. So now the question is why these deacetylases are being activated. So we don't know exactly what's going on. So we know some part of the answers, but you start answering, you find another player and you keep going.
Yeah.
But for example, in that combination we did with KRAS activation and primary cilia loss, KRAS can activate Aurora A, for example. Aurora A can activate HDAC6. So there are some oncogenes that may be the responsible to remove cilia. So for the idea we are presenting, primary cilia will likely be a second hit or a consequence of oncogenic transformation that helps the tumors to grow faster and migrate.
Okay, really interesting this. Okay.
Okay, let me
yeah.
finish one comment for the future. Hasta la victoria.
Siempre.
Siempre. Then,
Bueno,
Thank you very much.
no more question. Grazie.
Very good.
thank you so much.
Muchas gracias.
Grazie mille.
Ciao, grazie a te.
Ciao.